Hey guys, Mr. Hyatt here. This is part 3 of the Chapter 8 AP Biology lecture. So we've just got a few slides to get through here. Enzyme inhibitors, remember we're talking about things that impact enzymatic function. So enzyme inhibitors are going to reduce the effectiveness of enzymes. We've got competitive inhibitors that are going to bind to the active site. They're going to mimic the substrate. They're going to inhibit the ability of that enzyme just because the active site is full. Sort of like, guys, you've experienced this. Ladies, I'm sure you've experienced it too. You walk into the bathroom and all the stalls are full, all the urinals are full, whatever. You can't bind. You can't do what you need to do because there are no active spots to do what you need to do. Non-competitive inhibitors are going to bind to another part of the enzyme and change the shape of the enzyme to make it non-functional. So instead of filling up all the toilets, I'm going to remove them or something along those lines. Here's kind of a picture that demonstrates that. We've got here's our normal binding with our active site and our substrate. Well, there's a competitive inhibitor that binds here and doesn't allow the substrate to get into there. They're just physically blocked. And the non-competitive inhibitor is going to bind elsewhere, change the shape. Still, the substrate can't bind. Next up is allosteric regulation, and that's where that's basically our control mechanism, where we don't want an enzyme to be active all the time, so we're going to bind a regulatory molecule to it. We could have positive regulation, we could have negative regulation. We could turn the enzyme on, we could turn the enzyme off. Here's an example that shows that. Excuse me, we've got this activator that's going to cause the shape to change a little bit and is going to activate the enzyme. Or we've got an inhibitor that's going to bind and change the shape and make it non-functional anymore. That's a really important idea on the test. That's an important idea in the overall AP curriculum. So in terms of controlling metabolism, like I said, we turn our enzymes on, we turn our enzymes off. A couple of different ways that we do that are feedback inhibition and in structural order. Feedback inhibition we've talked about a few times. I'm talking about negative feedback here. So when a metabolic pathway is switched off by its end product, we call that feedback inhibition. Kind of the idea, we need something. So we're talking about an anabolic process here usually. So we need something, so we're making it. Well, once we have enough, we don't need to continue making it, so we get negative feedback that turns that process off. Here's kind of a picture that shows that. We get our initial substrate that's threonine. It binds to the enzyme complex. We change it through 5 enzymes. Then we make isoleucine. Isoleucine binds allosterically and changes the shape of the active site, changes the shape of the enzyme, so it can't work anymore. It can no longer function. Here's a couple of examples. Make sure you know these for the test for sure. Protein kinases are going to catalyze phosphorylation. They're going to make phosphorylation happen faster. That's going to be a really big one when we talk about cell communication. Protein phosphatases go the opposite way. So again, these are all going to be really important as we move through this semester. In both cases, the change in the protein's overall charge causes a shape change, negative-positive interactions, negative-negative interactions, which can activate or deactivate the protein or the enzyme. When we talk about structural order, what we're talking about is essentially separating these pathways in different places of the cell. So the example here is the enzymes of respiration. So those happen inside the mitochondria here. They're going to be in specific places inside the mitochondria. They're going to be on specific spots in the inner folds of the membrane. So that structural order controls our metabolism. So we can't do respiration. We can't break down food and turn it into ATP until it gets into the mitochondria. So again, it's not always going 24/7. So in summary, here are the 3 big points. This is really the beginning of our discussion of cellular respiration. The big thing here is enzymes, how they work, what the different types of regulation are, the active site, the induced fit model, all those types of things are the big things that we need to take out of this chapter. You probably know the two laws of thermodynamics, and you probably know the role of ATP in cell energy just from the fact that you've been through biology, you've been through chemistry, or you wouldn't be here. So that's chapter 8.